Thank you for clicking on my poster, which is about integrative modeling of chondrocyte mechanotransduction and biochemical stimulation. Osteoarthritis is a disease that affects older people, and its management only deals with symptoms because their mechanisms are not fully understood. However, we know that chondrocytes, the only cell type of cartilage, become unbalanced towards a catabolic state due to overloadings or pro-inflammatory cytokines. However, we do not know how these mechanic factors are coupled with the biochemical complexity, and in order to deal with that, we will apply network-based systems as a computational approach. Network-based models are a powerful framework to study signaling pathways within a cell. First, a static graph must be developed. There are different strategies used to construct it, but we use a knowledge-driven approach. Our network is developed by summarizing the biological knowledge into a static graph, where each node of the graph is a molecule, and they are connected to others with activating and inhibiting links. Then our static and integrative graph is built. In order to obtain dynamic behaviors, the static graph is translated into a system of ordinary differential equations based on network topology. The system, when it's solved numerically, it reaches a steady state, which can be extrapolated into a cell state or cell phenotypes. Indeed, steady states can change according to the imposed initial conditions. Then, they become a useful tool to explain dynamic behavior under different conditions, and that is why they are useful for calibration and validation purposes. The first step is summarizing biological knowledge into a static graph. So, we have created 52 articles. Then, the static graph is translated into a system of ordinary differential equations in order to obtain dynamic behavior following Mendoza et al. Methodology. The system of ordinary differential equations is solved through Runge-Kata-4, and a steady state is reached according to the imposed initial conditions. We have tested three different initial conditions: a physiosmotic one, a pro-inflammatory environment, and a non-healthy mechanic initial condition. The steady state reached can then be extrapolated into cell states or cell phenotypes. Finally, the model is evaluated qualitatively because we don't have any experimental data available. So we have established a research strategy to look for peer-reviewed articles in PubMed. Then we have reproduced the conditions found with our model. Here it can be seen the final static graph with 108 nodes connected through 237 links. If we take a deeper look into the results section, we can see in Figure 1 three different steady states according to three different imposed initial conditions. The first steady state that our network goes when healthy initial conditions are imposed is a healthy one because structural proteins, aggregain and collagen type 2, have high expression rates, as well as anti-inflammatory cytokines and healthy growth factors. But when inflammatory or overloading mechanic initial conditions are imposed, our network shifts into totally catabolic final steady state, because we can see higher expression rates in proteolytic enzymes, pro-inflammatory cytokines, and BMP2. The same behavior can be observed in transcription factors, because SOX9 is a transcription factor deeply associated with a chondrogenic phenotype, and it's only expressed when a healthy initial condition is tested. Then, when these unhealthy initial conditions are imposed, SOX9 becomes unactivated. The same happens with SITE2. And the contrary happens with NF-kappa beta, RUNX2, or AP1, which are transcription factors commonly associated with osteoarthritic phenotypes, or inflammatory cells. Then we test our model against a qualitative evaluation. To that, we summarize well-known chondrocyte responses with different loadings, and we test these loadings by activating or inhibiting the associated mechanosensors. When we do that, we can see that our model could represent the 60% of the responses tested. However, as it is not a really high percentage, we aim to calibrate our model against experimental data. As main conclusions, we can say that our model can integrate metabolic switches well according to the initial condition imposed. It can also reproduce 60% of the literature-based behaviors found, but further topology is needed against experimental data. Well, thank you very much for listening. I will be happy to answer all your questions. And first, I would like to acknowledge our sponsors and our founders. And here you can see our department, and we are placed in Barcelona. Thank you.